Hello viewers, today I will discuss about the structure and properties of the enzymes. Enzymes and life processes. The enzymes are biocatalysts which accelerates the rate of reaction. Plant as well as animal enzymes, they regulate homeostasis. They maintain life processes. The assay as well as pharmacological regulation of the enzymes are very much needed or the key elements for diagnosis as well as for therapeutics. Kuhn first coined the term enzyme for yeast ferment. Then he named trypsin, pepsin for animal systems. Intracellular enzymes, they regulate the internal metabolic pathways. The plasma membrane enzyme, after receiving the external signal, they regulate the cytosolic activities. Plant enzymes. The structural elucidation, the three-dimensional structural elucidation of plant enzymes were actually started from the beginning of 2000. 140 plant proteins were characterized via structural elucidation, among which 37 were plant enzymes. In recent 5 years, the structural elucidation or characterization of plant enzymes are done via X-ray crystallography or NMR study. The domains, the three-dimensional domains for plant enzymes and animal enzymes are very much conserved according to its catalytic process. The structure of enzyme. There are two main points, that is active site of enzyme as well as coenzymes and cofactors. Now active site is a three-dimensional pocket, it's a three-dimensional cleft where according to the size, group as well as form of the substrate can be accommodated for catalytic purpose. So it's a site for substrate binding. Sometimes the catalytic triad is important. Now some specific amino acids configuration and conformations are very important for the construction of active site. As we know, in case of serine enzyme, for acetylcholinesterase, serine amino acid is present. In case of papain, thiol containing cysteine is present. In case of transaldolase, lysine is present. Epsilon amino group of lysine is very important for accommodation or binding with substrate as well as for catalytic processes. So it's a kind of three-dimensional structure made by the protein folding and constructed by some specific type of amino acids. Now what is catalytic triad? Catalytic triad is important for some of the serine proteases. Say for example, in case of chymotrypsin, three major amino acids, they are important according to structure as well as configuration and conformation to bind to specific type of substrate. They are aspartic acid, histidine and serine. Now for plant enzymes, say for example beta-glucan endohydrolase, they can accommodate various types of oligosaccharides by their active site cleft. There are two types of active site models. One Fischer template and another one is Koshland induced fit model. The first one, Fischer's template or lock and key model. So active site of the enzyme is pre-made, it's somehow rigid. It is already made template to accommodate and to bind to the substrate. Okay, so the 3D structure, this is quite rigid, so it can directly bind to the substrate like the lock is fitting with the key. But after binding to the substrate, the product is getting released. This particular model does not explain all the properties of the enzyme, fine, but it obeys stereospecificity and some other aspects. But allostericity, the competitive inhibitor binding, they are not yet explained by this model. Okay. The second one, Koshland's induced feed model. It is somehow flexible, right? So already pre-rigid template is not yet made. When substrate approaches, according to the structure of the substrate, the conformational changes happen. Then ultimately, fully active and complementary conformation is made from loose to tight binding accept. Hexokinase is an example. So by this way, the allostericity as well as competitive inhibitor binding, some of the aspects or function of the enzymes can be well explained. So it is somehow the flexible active structure, active site model as well. Coenzyme and cofactors. So enzymes are basically protein in nature, but they need some helper molecule for their activity. So that is called coenzymes. not coenzymes, actually cofactors, they have some aspects of the division. The types of the coenzymes or cofactors, they are mainly inorganic or organic. The inorganic cofactors may be metal ion or iron sulfur cluster. Iron sulfur cluster, they are actually present in case of electron transport chain, where we can find the presence of cysteine residues which are contributed from the protein part. And organic, they may be coenzyme or maybe the prosthetic group. Now, what is the difference in between coenzyme and prosthetic group? Prosthetic groups are tightly bound with the enzyme, but coenzymes are loosely bound. Say for example, thiamine pyrophosphate. It is coenzyme for pyruvate dehydrogenase enzyme, but it is acting as prosthetic group for pyruvate decarboxylase. Now, pyruvate dehydrogenase is an enzyme where thiamine pyrophosphate is acting as coenzyme, but FAD, it is acting as prosthetic group. Usually, these types of coenzymes and prosthetic groups, they are bioactive version of the vitamins. Maybe sometimes some non-vitamin organic molecules are there, like S-adenosyl methionine. They are involved in case of one carbon transfer. The vitamin portion of the coenzyme or the bioactive form of the vitamins which are served as the coenzyme or prosthetic groups are several. Like in case of niacin, it is the vitamin, the coenzyme form is NAD plus or NADP plus, which is involved in oxidation reaction. Pantothenate is the vitamin and coenzyme A is the coenzyme version. So, it is involved in acyl group transfer. Biotin is the vitamin and it is acting as coenzyme in case of carboxylation reaction. Vitamin B6, where we can get in case of transaminases for transamination reaction, they are acting as pyridoxal phosphate as well as pyridoxamine phosphate. So, they are all bioactive version of vitamins which are acting as either coenzymes or prosthetic group. Next one, there are so many metal ions like copper, Cu2 plus, which is acting as cofactor for cytochrome oxidase enzyme. Iron, it is acting as cofactor for catalase and peroxidase. Magnesium for hexokinase. Manganese for arginase. Nickel for urease. Selenium for glutathione peroxidase. Zinc for alcohol dehydrogenase as well as carbonic anhydrase. But they are quite usual in case of our system. Some of the metal ions which are acting as cofactors, sometimes they are toxic for us, like vanadium. It is the cofactor for nitrogenase enzyme in case of azotobacter. Tungsten, which is acting as the coenzyme for enzyme aldehyde oxidoreductase in case of Pyrococcus furiosus archaebacteria. Cadmium, which is the cofactor for carbonic anhydrase for the system Thalassoria vis flogi. General characteristics of enzymes. Enzymes are usually protein in nature, but sometimes simple protein, only protein part is present in case of urease. But in case of catalase, we can find it's not simple protein, it's complex protein, along with which the metal ion is associated. Single polypeptide chain, it is found in case of DNA polymerase 1 for E. coli, but for pyruvate kinase is oligomeric protein, more than one subunit is present. In case of citrate synthase, it is homodimeric. Yes, two subunits are present, but they are identical. In case of mitochondrial ATP synthase, that is heterooligomeric protein, because all are not identical. The evidences to prove the protein nature, obviously, because they can raise antibody, having amphoteric nature, they are regulated via pH and temperature. They are all the evidences, yes, usually the enzymes are protein in nature. Now, the similarities with the inorganic catalysts. They need minute quantities which are getting associated with the substrate, but both of them cannot initiate any reactions. They can only accelerate the reactions which have already started spontaneous and which are in the equilibrium, and after that they are getting released by releasing the product. They are the similarities, but differences are still there because enzymes have specificity which is lacking in case of inorganic catalysts. They are regulated via pH, temperature as well as genetic control. All those enzymes, they are having higher molecular weight, but inorganic catalysts, they are having very low molecular weight. Some of the enzymes, they are getting secreted outside. In case of bacterial as well as fungal enzyme, for their digestive purpose, nutritive purpose, they are getting secreted outside. Fine. But so that's why they are called exoenzymes. But in case of plant enzyme like polyphenol oxidase, they are actually surrounded within the intracellular compartments because the metabolism is going on there. They are called endoenzymes. The colloidal nature, chemical nature, catalytic nature of effectiveness, the specificity of the enzymatic actions, thermolability, pH sensitivity, as well as reversibility of the action or reaction. Colloidal nature. The enzymes are globular. They have compact structure having high molecular weight. Say for example, pepsin 35,500, catalase 250,000. So, the diffusion rate is quite low for the enzymes. Chemical nature. The protein part of the enzyme is called apoenzyme. But when it binds with the coenzyme or the prosthetic group, the holoenzyme is made, which is actually effective to bind to the substrate, to accommodate with the substrate as well as to release the product. Now, catalytic nature of effectiveness. Enzyme is very specific according to its action, and there are so many enzymes having higher turnover number. Turnover number means the amount of substrate molecule to which it reacts per second. For lysozyme, it is 0.5 turnover number per second. For carbonic anhydrase containing zinc, the highest turnover number is present. Enzyme specificity. The absolute specificity, that means the enzyme is very specific to react or to drive a specific type of reaction by reacting with specific types of substrate. So, reaction specificity as well as substrate specificity. In case of plant enzyme, beta-D-glucosidase, they are strict about binding with the substrate. But in case of beta-glucan exohydrolase, they are quite flexible. Now, when we are talking about the substrate specificity, always there are 4 types. Number 1, group specificity. 2, chain length specificity, 3, optical specificity, as well as geometrical specificity. In case of group specificity, the enzymes which are attacking the glycosidic bond, glycosidases, they are so much specific. Chain length specificity, in case of fatty acid metabolism, they are very specific with respect to the carbon number. In case of acyl-CoA dehydrogenases, they are very specific with respect to the carbon number present in fatty acids. Optical specificity. It is all about the DL stereoisomers. So, we know that D amino acid oxidase, they are specific to react with the only D amino acids. Geometrical specificity, cis trans isomer specificity. As we know, cis aconitase is only applicable over the cis isomer. Next one, thermolability. Enzymes are very sensitive to heat. Rate increases when temperature is getting increased. But if we follow the activity of the enzyme as well as the temperature, there are 2 the factors responsible. 1, so when the temperature is getting increased, usual increase due to the increase of the collision. So, rate is increased. But after some times, we can follow the decreased rate due to the thermal denaturation of the enzyme protein itself. So, ultimately it is a curve, a bell-shaped curve when we consider 2 factors together. So, there is always an optimum temperature, so where we can get maximum catalytic activity. For usual enzymes, it is 37 to 40 degrees centigrade. So, if we can follow within the temperature optima, so there is a reversible reaction in between the active as well as inactive version of the enzyme conformation. So, we can go back to the original conformation as well as active state of the enzyme, but within the temperature critical limit. But if it goes beyond that, then the enzyme becomes coagulated. We can find in case of ribonuclease as well, they are getting denatured as well as renatured, but within the temperature optimum limit. Fine. Now, it is found that enzymes are usually stored in between 0 to 4 degrees centigrade, because at that particular temperature, the enzyme activity is retained. At very high temperature, the enzyme is lost. Sometimes at the frozen condition also, the activity may be lost. But in case of some thermophilic bacteria as well as seed and spore, the higher temperature optima is found, maybe 85 degrees sometimes, maybe 100 to 120 degrees centigrade. So, we can find usually it is a bell-shaped curve where we can considering the enzyme activity versus temperature. Now, temperature coefficient. We can find for every 10 degree centigrade increase in temperature, the double or triple action of the activity is getting increased. So, that is called Q10. But it is actually happen within 0 to 40 degree centigrade limit. pH dependence. Enzyme is also very much dependent on the pH because, as we have already told, the configuration and the conformation of the active site amino acid is very important. Usually it is a bell-shaped curve, and we all know when we are determining the activity, the kinetics, the pH must be constant because the Km, Michaelis-Menten constant, can vary. pH optima in the in vivo system, so we know that pH as well as activity once again a bell-shaped curve. So, for the in vivo system, so there is one part of the graph is actually considered, and we can find the effects of pH on the enzymes. What sort of effects? Because the change in pH can change enzyme substrate binding capacity, because the state of ionization of substrate as well as enzyme is very important to make the transition state ES complex. Sometimes the change in pH can cause apoenzyme dissociation. It affects the redox state of the cofactor metal ions. At extreme pH, the tertiary and the quaternary structure of the protein is getting abolished. As well as at higher pH, some zymogens by the hydrolysis of the peptide bond become activated. So, these all are the pH effects. As I have already told, the activity versus pH curve is bell-shaped, but some of the enzymes are highly acidic. Pepsin has pH optima from 1.5 to 1.6. Acidic invertase 4.5 pH optima. Alkaline fumarase 7.8 to 8.7. Highly alkaline arginase, it is around 10. There are some exceptions for this bell-shaped graph. We can find in case of papain, if you can follow the activity versus pH graph, it's a straight line in between 4 to 8, because thiolate as well as imidazolium ions pair, which are important for its catalysis, only we can find within this range. In case of cholinesterase, the activity is actually highest in case of 6, then they are getting the plateau level. Once again, histidine must be present. We can find in case of trypsin, there is a long range 2.5 to 11, because below 2.5, we can find the active carboxyl group will be protonated version, right? And beyond 11, we can find once again the epsilon NH3 plus will be in the NH2, which are not needed. So, for its activity, we needed COO minus as well as epsilon NH3 plus, which are having within this range. Once again, if you follow in case of pepsin, we can follow it is 1.6 to 3.2. In this region, we can find two carboxyl group. One is in COO minus conformation, another one is in COOH conformation. Both of them, both the kinds of carboxyl group we are needed for substrate binding as well as interaction. So, enzyme can drive both the forward as well as backward reaction. But the thing is that it is dependent on the pH of the cell sap or the environment as well as availability of the reactants and the end product which are getting produced. So, reversibility is actually maintained in case of lipase by all those actions, which is actually important to hydrolyze fat to produce fatty acid as well as glycerol. Now, so we are talking about the structure and the properties of the enzymes. So, there is always structure-function relationship. So, bonding to retain the structure of the enzyme and substrate is very important. Superfamily of the enzyme is important, as well as active site amino acids which are taking part in the catalytic processes are important. Now, bonding. To make the proper bonding to follow the alternative pathway in between enzyme and substrate, van der Waals interactions, hydrogen bonding, ionic interaction, hydrogen bondings are made. Now, the superfamily. So, what is superfamily? There are some specific fold within the structure. So, sometimes maybe we can find the similarities in the amino acid sequence or may not, but they are driving same types of reaction. So, we can find allozyme, isozyme, etc., but they belong to the main superfamily according to its action. And according to the presence or absence of different types of amino acid, we can find either acid-base catalysis or covalent catalysis, metal ion catalysis, so many different types of catalytic strategies are present. In future, when we are talking about the plant enzymes which can regulate plant metabolism as well as physiology, so there are some modifications can be done by bioinformatic analysis way. So by which if we ascertain the domain of the plant enzyme, they can be taken for any further processes which can encourage the plant cell biology as well as molecular modeling processes. So protein inhibitor binding, the docking of the phytohormones to a specific type of receptor, they can explore more and more opportunities for increasing plant productivity as well as product quality, which are very much needed for the molecular evolution. So that can be done for no more new aspects in case of plant physiology and metabolism. So students, we have already discussed about the structural aspect which is tightly regulated and controlled by the functions of the enzyme as well. We have discussed the active site of the enzyme, the definition, the models of the active site, the catalytic triad. We have discussed two models, Fischer template as well as Koshland induced fit model. We have discussed the helper coenzymes as well as cofactor which may be organic or may be inorganic in nature, the types. I have mentioned some of the vitamins whose biological conformation is needed for the enzyme activity. I have discussed about the metal ions which are acting as cofactor for several enzymes, the general characteristics of enzyme, that is enzyme binding to the substrate making transition state which can help to lower down the active energy of the system, as well as whether they are single protein or multimeric or oligomeric, whatever. The evidences for being protein in nature, similarities as well as differences with the inorganic catalysts. I have discussed about the exo enzyme as well as endo enzyme. Exo are secreted outside, but endo are confined within the system, within the cell itself for the sake of the metabolic purposes. The general and the physicochemical nature, colloidal nature, chemical nature of effectiveness, specificity, that is most important for enzyme action, thermolability, pH sensitivity as well as reversibility of the reaction. Among them, the pH sensitivity and thermolability is very, very important because we know for in vivo system there is a certain amount of energy budget, right? So enzyme action must be regulated by the temperature available there. If the curve is bell-shaped, whatever, so the action may be on either side of the bell-shaped graph. That is also applicable for the pH because there is a certain kind of constant in vivo pH, say for example 7.4. So though it may have a certain pH optima, but the action must be regulated by the pH present in the in vivo system. Okay, so they might be on either side of the bell-shaped graph. And I have discussed about the pH dependence as well as I have discussed about the reversibility. Obviously, the reversibility of a particular enzymatic reaction is regulated by the ambience, okay, as well as the presence of reactants and the product. The structure-function relationship which are tightly regulated by the homologous superfamily because when they belong to a superfamily, obviously some homologous folds are present, as well as the presence or absence of some of the amino acids which can regulate different kind of catalytic processes, the bonding involved to make the enzyme substrate complex. So in future, we can go for many more three-dimensional modeling aspects by bioinformatic analysis for phytohormone targets to regulate the specific plant metabolism as well as physiology. Thank you.